G'day guys, this is a video on leucine and its effects on cancer. Let me just share my screen. Okay, so this study was published in the 29th of October 2019. Leucine rich. So this was in Nature. So it's not some, so this is one of the top magazines or periodicals in the science literature. So, most people hope in academia that they get published in Nature. But anyway, let's move on. So it's not some bullshit study. Leucine-rich diet induces a shift in tumor metabolism from glycolytic towards oxidative phosphorylation, reducing glucose consumption, metastasis. So we know what metastasis is, when, the cancer proliferates throughout the body. In Walker 256 tumor-bearing rats. So obviously it is in an animal model, because you can't do this research in humans, unfortunately. They don't go through ethics committees putting people on interventions of this sort. It's unfortunate, but that's the reality. But leucine via mTOR in the musculoskeletal and stuff like that works very similar in all mammals. So it's something that's been preserved genetically throughout all mammals. So it works in a very similar way. It's not like a diet. A diet can be species specific. So if you can't do a dietary study on rats and say that then you can extrapolate that to any other animal, any other species. That's just nonsense. And that's why the majority of the diet studies I've never shown on this channel, I never will, because they're just nonsense. The only stuff that I have shown is sort of whether a piece of food causes some oxidative stress or not. And that's an interesting thing, but it's also can be a bit on the irrelevant side. But in this case, what they actually noticed is in the research that a leucine-rich diet induces a shift away from, a sort of a phenotype of glycolysis away to oxidative phosphorylation. And that's critical. And I've been talking about this, how to shift cancer cells away from either back to senescence or to cause apoptosis and get rid of them. And why my intervention of protocols, which combine a carnivore diet, which is a leucine-rich diet, and also has taurine, has choline, it's also a choline-rich diet, and all these things do have positive effects in terms of improving people's survivability, reducing sort of wasting away, and a whole lot of other conditions which really work against people and usually weaken them so much. And let's not forget, the immune system requires a whole lot of amino acids, while vitamin D and taurine play an important role in stem cell production and then activation and regulation of immune stuff. We still need the material to construct the stuff. Without having enough protein in our diet, we can't build. They're the building blocks, the amino acids. So let's move on. Oops. So leucine can stimulate protein synthesis in skeletal muscles, and recent studies have shown an increase in leucine-related mitochondrial biogenesis. That's really important. And oxidative phosphorylation capacity in muscle cells. So there's a number of studies, not only this one. This is looking from an angle from a cancer model, but there are a lot of other sports physiology type ones that actually, more recent ones that are actually looking and realizing that leucine is actually causing biogenesis of new mitochondria. This is probably one reason why people's energy levels improve on a carnivore diet, and their general ability to heal faster. The more energy you can produce because you've got more mitochondrial density, the better you're able to basically heal faster. So, leucine is playing a critical role in this effect, but through the signaling of certain pathways. We'll get into them soon. However, leucine-related effects in tumor cells are still poorly understood. They are. There hasn't been much. Most of the stuff out there is garbage. Most of the stuff tends to be epidemiological, and it tends to be people on sad diets, eating sugar and a whole lot of other crap, driving mTOR in the fat cells, not in the muscle cells, but in the fat cells, because that's what glucose does, and increasing fat accumulation and also causing other derangements in the body, which are related to Randall cycle inflammatory pathways. So up until recently, leucine has sort of been known as an important amino acid for the signaling, the mTOR signaling in the musculoskeletal, muscles and bones, to sort of drive protein synthesis. But it hasn't been looked in terms of other roles that it plays in the body. And I'll be covering a number of these roles in future videos because they do affect the brain and other tissue, so mental health and a whole lot of other things as well. And it sort of explains why a number of people are getting improvements in mental health, like bipolar, schizophrenia, and a whole lot of other things. But that's for another video, a future video, to cover the mental health side and important aspects of leucine. Thus, we describe the effect of leucine in both in vivo and in vitro models of Walker 256 tumor rats. Tumor-bearing whisker rats were randomly distributed into a control group, W, and a leucine group, LW. So just remember that, which was 3% leucine. After 20 days of tumor evolution, the animal underwent fluido-oxyglucose pistrone emission computer tomography. So, scanning, basically PET CT imaging. After euthanasia, fresh tumor biopsy samples were taken for oxygen consumption rate measurements, like electron microscope analysis, and RNA and protein extraction. We're not going to cover in this video because there's shitloads of pages in this. So we're going to go to the key sort of findings and all that soon. Now, our main results from the leucine group showed no tumor size change. So there wasn't lower tumor glucose uptake, which means that the tumors were struggling, and reduced metastatic sites. That means we we saw less metastasis. So metastasis was inhibited. That means proliferation throughout the body, which is important because that's what kills you in the end, metastatic cancer, when it metastasizes and just proliferates like mad throughout the body. Furthermore, leucine stimulated a shift in tumor metabolism from glycolytic towards oxidative phosphorylation. This is important. I've talked about this before, how shifting a cell from a cancerous state back to a senescent state, or if it's not viable, causing apoptosis to get rid of it. I've talked about this, how taurine plays this role, but also leucine plays this role as well. So really important. And this is why the combination of the diet and some of these supplemental things amplify these effects. And that's what we're trying to get, So high mRNA and protein expression of oxidative phosphorylation components, that's a good thing. And enhanced mitochondrial density and area. Even through the leucine-treated tumor had a high number of apoptotic nuclei with increased oxidative stress. That's a good thing. What do you think chemo does? And then you get chemo resistance, so that's pretty much those sort of cells are forced into cell death in a sense. So they'll find in these rats more areas that are just dead cancer cells when they were doing, we'll look at it further down. In a summary, leucine-rich diet directed Walker 256 tumor metabolism to a less glycolytic phenotype profile in which these metabolic alterations were associated with a decrease in tumor aggressiveness. That's really important. We need to pull back and reduce the aggressive growth of cancer, and reduction in the number of metastatic sites in the rats fed a diet supplemented with branched chain amino acids. Got it? So, and they just cover this. Cachexia affects up to 80% of advanced cancer patients, responsible for almost 30% of all cancer-related deaths. Cachexia related to cancer, termed cancer cachexia, is a multifaceted syndrome that leads to an involuntary weight loss associated with poor quality of life, poor prognosis, and high mortality. Now you know why I insist people go on a carnivore diet to maintain their musculoskeletal structure and it doesn't fall apart. The main cause of weight loss is skeletal muscle wasting. That leads to muscle activity impairment. That's what it is. That's a killer. And unfortunately, when people go into the medical system, what do they get put on? Kibble. Protein deficient diets. And we've got also people promoting protein deficient diets because they fear mTOR. mTOR isn't the problem. mTOR activation inside the musculoskeletal stuff isn't the problem, and it's signaling in there. The problem is in the wrong parts, pushing growth pathways, like in the fat cells. Thing that does that type of mTOR signaling is sugar. Thus, in order to prevent or treat skeletal muscle wasting, some nutritional strategies have been used. In this regard, leucine supplementation has been investigated due to its potential to increase protein synthesis in skeletal muscle. And I've actually have shown in the past video that I will attach, where even in a fasted state, if you put enough leucine in, you can actually get some level of protein synthesis, or at least, compared to somebody that doesn't put any. So it has a powerful signaling when it comes to the musculoskeletal. Alone or together with other branched chain amino acids. So on its own or in combination, which is the key ones, valine, isoleucine, and leucine has stimulatory action on skeletal muscle protein synthesis by acting as a cell-signaling molecule, leading to the activation of mechanistic targeting of rapamycin, mTOR pathway, and key cellular protein synthesis pathway. Moreover, leucine can also act as energetic molecule once it is a ketogenic. It is one of the actual, together with lysine, it's purely, there are others, but they can be both ketogenic and and gluconeogenic as well, the other like tryptophan and stuff like that. But lysine and leucine are purely ketogenic amino acids. So people don't need to worry about if some of the extra creates a few more ketones. That's fine. That's a bonus in a low carbohydrate state. So if you supplement additional, you don't really have to worry. The additional stuff will just can be used as ketone bodies if it's not used for protein synthesis, is what I'm trying to say. So can be useful. Anyway, moreover, leucine can also act as, we'll cover that. And also finally used in the citric acid cycle, the TCA cycle, so to produce ATP. Got it? So it's involved in energy production. Furthermore, leucine increases peroximone peripheral activator receptor gamma, PGC1 alpha, suppression leading to mitochondrial biogenesis. Got it? So that's the pathway. We know that actually if you fast, PGC1 alpha goes up if you fast, which is involved in mitochondrial biogenesis. And leucine does exactly the same thing, because it is, again, you'll see further down, you'll understand why there's a lot of confusion about leucine and improving oxidative metabolism in skeletal muscles. Exactly. So, oops. So you can see the growth. There's more growth, there's less growth in the rats on the leucine. Okay. As I said, people can, at their own, you can see PGC1 alpha going up, COX5A going up. So you can see these improvements in some of the other signaling pathways. So I'm not going to get into them. I'm just showing just generally. Leucine reduced glucose consumption and lactate production, and increased oxygen consumption, which is a good thing in a cancerous state. Got it? We want lactate to be down. The other thing that helps with reducing lactate is taurine. So the taurine-leucine combo is really good to reduce lactate levels. You know, very helpful to the cancers, lactate. And so those are the sort of reasons. Let's move on. And oops. So this is just the oxygen consumption increasing. Just to show that there's this increase in oxidative phosphorylation pathways. And they've seen it in both vivo and in vitro. So basically in the petri dish and in a species. It just pushes that shifting from one to the other. Really important. Okay, so we can see the different sizes. Again, so those who want to look at diet led to mitochondrial biogenesis. Okay, so we're seeing an increase. So there's one site, and there's multiple sites where there's increased in mitochondrial. People can read it when they get it. And so when they did biopsies, they also saw an increase in the density and how much area was taken up. So more area was taken up, which means that there was more mitochondria around, which is a good thing, because you're shifting away from fermentation, which is what cancer really likes to use, and you're shifting towards oxidative phosphorylation, which is what normal cells use, producing ATP in large amounts from, the mitochondria. That's what we want to see. Okay. Cell viability, looking at stuff like that. Anyway, there's a really important superoxide dismutase. You can see the differences. It's much lower. That's, at the initial stage. That's after they added something. Okay, let's get down here. Okay, in this study, we investigated the effects of branched chain amino acids, leucine, on the Walter 256 sarcoma metabolism. The main finding were a less aggressive tumor and a lower uptake of lactate in the presence of, and glucose, obviously, in the presence and induction of metabolic shift from glycolytic towards oxidative phosphorylation. Really important in both vivo and in vitro. So both showed the same behavior. When you added leucine, the cells did the shift. Most cancers, cells present high rates of glucose uptake and aerobic glycolysis. Anyway, I'm not going to go into the three-geared system. A phenomenon known as a Warburg effect. This phenomenon is observed in the majority of cancer cells and is considered one of the hallmarks of cancer, even though it is inefficient regarding ATP production. Aerobic glycolysis provides many advantages, such as rapid and easily accessible sources of glycolytic intermediaries that can be shunted into the pentose phosphate pathway. pentose phosphate pathway is a major glucose it's not me saying it, it's the research, is a major glucose metabolic shunt that is upregulated in cancer cells. Now, that doesn't mean the pentose phosphate pathway is a bad thing. It's just basically it's upregulated in a cancerous state. You know, what they see. But we see the opposite when you introduce. And also nucleotide synthesis, which basically we're talking about having to do with the DNA and all that. So producing more generating more cancer cells from being able to produce, subdivide the cancer cells. That's what we're talking here. Both of which are essential for cancer, for cells with a high proliferative rate. Don't you love my dyslexia? So they proliferate quite a bit. Moreover, this aerobic glycolysis causes an increase in lactate production and export. We know from a reverse Krebs cycle. I've talked about it before. I'm not going to go into it this time. Leading to the acidification of the extracellular environment, favoring metastatic formation. Yes, we know. That's one reason why you tend to urinate out more alkaline. It's a sign of cancer. When you're actually urinating out more, you're more acidic, it's actually a good thing on an animal-based diet. When anybody's urinating more alkaline, that's a problem that could signify cancer environment. And we're talking about very small amounts because otherwise you'd be dead. In this study, we found a decrease in 18F glucose uptake and lactate exportation, lower expression of LDHA protein in both the tumor biopsies and the leucine treated cells. Exactly. As you can see, it is really making it difficult for cancer cells to use their preferred fuel, so to speak. Consequently, we found lower metastatic sites in tumor-bearing rats fed with a leucine-rich diet. Exactly. And what is all the epidemiological nonsense out there that the vagoons and everybody else peddles? Oh, animal foods, rich in leucine, are going to cause cancer and kill you and all that. Bullshit. Absolutely nonsense. Anyway, it's you guys that have been following me now for a long time know that mitochondrial damage from deuterium and high oxidative stress from seed oils and all this other crap is what over time really erodes the health of our cells, and then we shift to a state where we can actually get into a cancerous state. But you guys are much wiser than most of the general public out there that listen to the nonsense of the epidemiological stuff, which you can't, determine anything because it's so confounded and so poorly controlled as research to be pretty much irrelevant. This is actual proper experimental stuff. We give this to one group we've induced cancer in these animals, and we give one group this and another group that. What happens? I wish they would do a leucine study on humans with cancer. Two groups, one with cancer, another with cancer, one with leucine, and another one without leucine. But hasn't happened yet. Those ethics committees, which are controlled by the National Institute of Health, Fauci's sort of domain, He's got from, well, people like Fauci type equivalents tend to basically say no to these studies. They've said no to the deuterium-depleted water studies and many other things, which is unfortunate for the American people and the global people that would like other options to be investigated. So we have to do studies on animals because the authorities will not fund these studies in humans. Anyway, this increase in oxidative phosphorylation capacity could be stimulated by acetyl-CoA, which is one of the final metabolic products of leucine metabolism, since acetyl-CoA is directly consumed by mitochondria through the TCA Krebs cycle. Leucine-treated cells favor oxidative phosphorylation instead of glycolysis. The oxidative phenotype found in tumor biopsies from the leucine group was observed in both phosphorylating ADP additives and non-phosphorylating cat additive mitochondrial uncoupling states, which reflect an increase in oxidative capacity of these tumor biopsies. Okay, accordingly, Walker 256 tumor biopsies and cells showed an increase in all bioenergetic parameters that were analyzed in the functional respiratory assay. Those were the oxygen stuff, which showed an increase in the ability to do oxidative phosphorylation to produce energy the way it should be produced. In agreement with these results, leucine treatment caused an increase in expression of mitochondrial genes COX-5A, CS, and ATP synthase. Okay, very important. We're getting complex. Remember that mitochondria, the nanomotor that gets damaged? This is it. What does this do? It actually improves, the production of ATP. Got it? Yes, a carny diet. Who would have thought? Proteins, NCS, and ATPase, which is complex 5, as you guys well know. You're well up to scratch on that. The complex through is usually where fat goes through, but remember, and so do ketones and all that. Complex 1 is where we get most sugar going through. Just remember associated oxidative phosphorylation in the tumor biopsies. These molecular data agreed with higher mitochondrial density and area as observed by TEM images in the leucine rat group. Oops. So, interestingly, leucine-treated induced metabolic reliance on oxidative phosphorylation, shifting those cells away. It's important, suggesting a direct effect of leucine on tumor metabolism. Exactly. A positive effect, not a negative one, which is associated with nonsense in epidemiological nonsense. This is experiments. We see mechanisms here. We see pathways being activated, gene expression, very important. Notably, our vitro findings show that leucine-treated boosts mitochondrial function. That's what we want, as indicated by increased oxygen consumption rates. So we know oxidative phosphorylation is happening. And reduction in lactate levels. It's a bloody good thing. That's what we want, reduction in lactate levels. We can add taurine to that mix and actually reduce it even further. In line with these results, PGC1 alpha, really damn important. What is it? I can't remember the actual thing. COX-5A and the other component expressions. These are all, there are genes for these that are expressed and transcripted, and they all sort of help improve complex 5 ATP synthase. Protein expression, that's what we want to see, were significantly higher in the leucine treated cells, suggesting that leucine treatment also activated the mitochondrial oxidative phenotype in vitro. So works in and outside an organism. Other studies have shown that leucine can increase oxidative phosphorylation mitochondrial biogenesis in other types of cells, such as skeletal muscles, and change the metabolic profile of these cells. Study performed by Bagan, Warburg, and colleagues evaluated, probably I've butchered that name, evaluated the effects of leucine treatment on oxidative and glycolytic metabolism in humans and murine rats, basically, skeletal muscle cells. The authors observed a significant reduction in glycolytic metabolism and also on lactate export in leucine-treated cells. Therefore, that group concluded that leucine could potentially induce an oxidative profile by increasing oxidative capacity in skeletal muscle cells. Bloody hell does. The blockage of lactate export may explain the alterations of a more oxidative phenotype. In previous studies, we found that lactate serum concentrations was increased in Walker 256 tumor-bearing rats in comparison to healthy rats. Yes, we know. This increase reflects high lactate production and its released by tumor tissue, reverse Krebs cycle, basically. That also means that glutamate can actually be used, that everybody worries about. Not a problem. It's only in reverse Krebs cycle where you can produce more, but we can use all these strategies to inhibit that so the normal cells can still get the glutamate and all the other stuff, because it's actually endemic throughout the whole body. There's no way of preventing cancer cells getting it too. They'll just migrate and infiltrate other neighboring cells to get their glutamate. So it's just, restricting doesn't really work. It just creates cachexia, muscle wasting, and all that sort of stuff. It accelerates that. It's bullshit. It's reductionist thinking, and it doesn't help at all. Leucine also causes a decrease in LDHA expression, favoring an oxidative phosphorylation metabolism in tumors in both vivo and in vitro experiments performed in this study, which is really, really good. So that just covers the same sort of stuff, talking about, let me find the other part. Okay. Besides metabolic shifting, we observed that leucine-rich diets stimulate oxidative stress and apoptosis. Exactly. So a meat a day will keep the oncologist away? Is that what we're talking about here? Yes, oxidative stress on cancer cells and apoptosis. Goodbye, cancer buggers. A high rate of oxidative phosphorylation production not only ATP, but also increases the oxidative stress. We need that. What do you think they use tumor against cancer to create oxidative stress to kill it? But it doesn't create extreme levels, which is important, and it targets, because normal cells can actually produce things like melatonin to heal the mitochondrial reduce, and also in the diet, things like taurine come in and actually reduce oxidative stress inside the normal cells. Not a problem. Don't have to worry. That's why, remember I was telling you studies years ago about, how they use to prevent chemo resistance, which means that people can't do it anymore because creating so much oxidative stress they can't bear it in the normal cells. So they get really sick severely. But with taurine, high dose of taurine, you can actually bear it because it protects the normal cells, but not and causes apoptosis and sort of amplifies the stress on the cancer cells. And that's what we want to do, kill the buggers. Anyway, furthermore, but the problem is with, as I said, with chemo and stuff like that, there is a problem with weakening the immune system. And the immune system is critical to basically fight off cancers. So we want to stress the little buggers to die, or shift them, or do the other thing. Anyway, now let's just continue this. A greater number of apoptotic nuclei, that means cells that are, being wiped out, was observed in the leucine group, in line with an increase in necrotic tumor mass. So what they found is a lot of the tumors were just dead, tumor mass. Eventually the body will clean all that up. There are mechanisms inside the body. If you've got necrotic tissue that's, eventually macrophage and all that move in the immune system, it cleans it all up, so you don't have to worry, But we want these buggers. The same thing happens when you do chemo. It creates these necrotic tumor masses, but we don't, without the risk to the normal cells. Wonderful stuff, old leucine. Observed in PET and CT images. Nakano and colleagues, so Japanese research, showed BCAAs enhance senescence in liver cancer. That's important. Remember what I said. We want to shift back to oxidative phosphorylation and shift those cancer cells back to senescent, or cause apoptosis, clear them out, which was stimulated by DNA damage mediated through mTOR1 pathway, increases P21 translation. That P21 is involved in DNA repair. Yes, DNA repair. And activated by P53. And P53 is also an important element in called tumor protein. It suppresses, that's right. This protein acts as a tumor suppressor, which means that it regulates cell division by keeping cells from growing and dividing, proliferating too fast or in an uncontrolled way. Got it? And leucine increases this bugger. And this bugger, as I said, does DNA repair. P21 allows DNA repair to proceed, blah, blah, blah. I'm not going to get into it. That's for another day, to go into mechanisms of how a lot of things that work within cancers. People want to learn that. Yeah, I may do some sort of future study thing, going through it and explaining all these different transcription factors that are transcripted to basically, run a whole lot of enzymatic processes to get things to do this, that, and whatever else. Anyway, activated, leading, and this is the important stuff, leading to senescence and apoptosis. So a cell will either shift back to a senescent state, downregulate the oncogenes, or if it's not viable, gone. Got it? Wonderful stuff. That's what we want to see. That's what we want to do. Taurine does the same thing. Shifts cells back towards a senescent state, improves oxidative phosphorylation as well. I've discussed it before. We can see here a gluconeogenic amino acid like leucine. Powerful stuff. Accordingly, we observed an increase in protein expression. We've covered that. Yep, the P. And also, we saw a decrease in the incidence of liver cancer in both obese patients and diabetic model animals of carcinogenesis. So animals that they induced on a sad diet, what they call a high-fat diet, which is really a sad diet, seed oils and sugar. And pretty much what they saw was an improvement in those models in terms of obesity, and that were either obese or diabetic and had cancers. They saw improvements, a decrease in the liver cancer. Anyway, so that was that part. I think that's pretty much it. I'll just cover this last paragraph. In conclusion, we described for the first time the effects of leucine-rich diet in Walker 256 tumor metabolism. Our data showed that leucine induced a metabolic shift from glycolytic towards oxidative phosphorylation in tumor biopsies and isolated cells. The metabolic shift was accompanied by enhanced mRNA and protein expression of oxidative phosphorylation components, okay, transcription of those things, higher mitochondrial density and area mass that they took up. So there was, moreover, these metabolism alterations were associated with a decrease in tumor aggressiveness. Tumor aggressiveness is what kills you in the end. If tumor can be slowed down, you're going to live. You've got a stronger ability to enhance, to use senolytics like Pfizer and all these sort of things to try and kill things off, improve your immune system, blah, blah, blah. Suggesting that leucine nutritional supplementation does not benefit this type of tumor. Fantastic. That's what we want to know. It is important to emphasize the interpretation of the results considering only the Walker 256 tumor metabolism. Obviously, this is, an animal model. We do need human trials. When are we going to get them? Don't know. No idea. But this is the first in an animal model where we've used leucine. In a good percentage, 3% is pretty good within a food component. So it's not like a small amount. And pretty much what this research is showing us is that there are novel strategies which are animal-based that have a lot of benefits. And we can see that mTOR is behaving very differently. There was another thing that I did want to cover. Let me just... That was related to mTOR. These are all the methods that they used. Let me just pause the video for a sec so I can actually try and find the damn thing. So, was this one down here, this actual study that actually looks at leucine modulates mitochondrial biogenesis, SIRT1 and AMPK. So this is one of the number 6 reference. Metabolites of leucine have been reported to activate AMPK in this case with resveratrol. That was that guy Michael Zemel, who's, I still haven't made that video. I need to make it on him, but it does synergistically work together with a number of other things and actually has this amplifying effect. But even in a fasted state, if you take leucine, it'll actually have an effect on amplifying AMPK. And in a low carbohydrate state, it still has some upregulation of AMPK pathways, which we know that is actually what happens also in, when you're fasting. So it does have certain synergies. And just to remind people that all the sort of reductionist nonsense that's out there, mTOR is a much more complicated thing. And leucine is a much more complicated thing. It's not what people tell you, because I'll say, oh, it's pushing mTOR up. Yeah, but it can actually, leucine can also affect AMPK. So it's not here or there. There's a lot of signaling that actually happens, that takes place in the body. It happens where? And so in the fat cells, I need to find that study. I can't remember where I've put it. But in the fat cells, what it does is it actually, mTOR actually increases sort of AMPK sort of signaling with leucine present. So immediately you're actually getting a completely, if you throw glucose in there, it'll do the complete opposite. It'll suppress AMPK and amplify, all those anabolic pathways within the fat cells. What you don't want, that's increasing inflammation. That's increasing, all these, which is all sort of the end result of, the activation of the Randall cycle. Pushing these sort of signaling molecules to signal in such a way and have this cascading effect in the different tissue types. And that's the important thing, that we need to realize the difference. When we're taking leucine on a low inflammatory diet without the sugar crap, what we're doing is we're pushing the signaling in the musculoskeletal to strengthen them, to increase them, to make them more viable. And we're also, any cells that are damaged or whatever, we force them into apoptosis, cell death, or we try and recover them back into a senescent state where they're not proliferating anymore as cancer cells. And then if you've got a good enough immune system, they should be cleared up over time. That's the strategy we want to use when we're fighting cancer. I've got a whole lot of, videos covering cancer, but this is important because a lot of people worry about, I'm doing a carnivore diet. Is it putting me in danger? As long as you don't put the sugar in there, boys and girls, no. You don't have to worry about it, because your leucine is going to be pushing pathways that suppress proliferation of cancer, that basically try and arrest cancer by inhibiting its growth, by pushing those cells back into a senescent cell or causing apoptosis, so cell death can actually be initiated. And this is why I'm saying carnivore diet with the senolytics, with taurine, with a number of these sort of strategies can actually enhance the clearing up and lowering the damage to using deuterium depleted water to eliminate the damage so we don't continue the mitochondrial damage. You know, these are the strategies that we can use. There's a couple of peptides and stuff like that that are involved in improving the immune system, which is really important, you know, so we need to sort of enhance, you know, the immune system. And that's critical. And that's, again, animal foods are basically, or components within animal foods are sort of helping those pathways. They'll be for a future video, and I'll be covering some of the actual signaling that's happening with leucine, with mTOR, and a few other things that actually have additional benefits, you know. So, and you'll be able to use this information and go, well, hold on, you know, Vagoon, you know, that's not the case. Here's an actual experiment. Yours is epidemiological nonsense, associated claptrap. You know, association does not mean causation. Here's an experiment showing specific transcription of specific, you know, enzymatic processes that are happening inside these cells. There is a whole lot of activity that is basically reverting and inhibiting that from going into a worse metastatic state, and it's pulling it back, either killing it off or pulling it back into a senescent state, which means it's reducing the level of tumorigenesis and tumor growths inside and creating more necrotic cancer cells in the body, which eventually the body will clear up, which is what we want. We want to kill the damn things and clear them out, and eventually the body will slowly but remove them over time. That's the strategy. These people are so caught up and fixated on associated nonsense, propaganda, basically, that they're providing really poor information to their subscribers and their supporters, which is unfortunate because some of them do have a number of health problems and are put at higher risk. I look at experimental evidence. I look at stuff which is solid, which actually shows mechanisms that show you take this molecule that has this effect, this signaling effect, and this is what happens. You know, here's a model, an experiment. We've induced cancer. Okay, it's a cancer model. You give leucine. The belief out there in the crazy world of epidemiology is that animal foods, and leucine in particular that drives mTOR, is a killer. It's not. This is the evidence for it. This is what you use to argue against that nonsense. Anyway, I hope you enjoyed this video. There'll be more like it. Take care.